Okay, so there's group 4, which is single-stranded RNA, and then there's a positive sense single-stranded RNA. This is an example of, for example, picornavirus that I mentioned previously. Hepatitis A virus, I believe, is also positive sense single-stranded. And the nice thing about this for the virus is that this is just like a positive sense single-stranded RNA virus. Basically has viral RNA, so it's right there. So it has viral RNA that can be used as messenger RNA. So proteins can start to be translated immediately, and the first thing the virus makes is an RNA-dependent RNA polymerase because it has to make more of its genome. And we'll talk a little bit more next time about how it makes its genome. So group 5 is a negative sense single-stranded RNA, and in this case, so the virus has to go through, it carries its own polymerase because it has to make a positive sense RNA in order to get, so it uses the negative sense RNA template to make a positive sense RNA in order to get to positive sense RNA. And then group 6 is retrovirus. Retroviruses are, an example here is HIV. They make reverse transcriptase, and that turns RNA into DNA, double-stranded DNA. Very famous virus, obviously HIV. And don't worry about group 7. We won't talk about double-stranded DNA pararetroviruses. Sometimes hepatitis B can fall into this category. Okay, so once again, the examples of viruses are here, group 1 through group 7. I'm not going to hold you responsible for group 7. I think I did not mention that in a very important negative strand or antisense, negative sense, antisense, same thing, single-stranded RNA virus is influenza, and we will be talking about influenza virus and its replication strategy. Okay, so viruses bind to receptor proteins on the surface of their host cells. So for every virus that has a viral receptor, like one of those spikes on the adenovirus, the one that looks like a satellite that has a viral spike, that's the viral receptor for a host cell receptor, and those interact with each other. So, and receptors determine the viral tropism. So most viruses are very, very specific and will only infect certain cell types. As an example of that, some bacteriophage, which remember are bacterial viruses, some bacteriophage will only infect certain types of bacteria. And people have taken advantage of that. What they've done is they say, well, we know that certain bacteriophages will only infect methicillin-resistant Staph aureus, or MRSA. Many of you have heard of MRSA. It's a problem in hospitals. So there's something called phage therapy or bacteriophage therapy where they're using a virus. When antibiotics have failed for patients, the bacteriophage goes in and infects MRSA and kills off MRSA. So things like that are in clinical trials right now, and they're looking pretty good, promising. Okay, so some cell receptors are lots of different host cells, but most have limited expression. Influenza doesn't infect every single cell in your body. So an example of a broad here is a rhinovirus, which is obviously a cold virus, binds to intracellular adhesion molecule. Primarily, though, gives you a cold, even though you have ICAM on a lot of different cell types. And HIV is another example. It's very narrow. It only binds to CD4 and a co-receptor on CD4 cells, or T helper cells. Okay, so you have to know this. You have to know what are the steps in viral replication. This is an objective. So you have to know that the first thing a virus has to do is attach to its host cell. So this would be looks like a non-enveloped virus here, and some viruses have spikes on them. This cartoon doesn't show spikes; it shows some indentations here that the cellular receptor is grabbing. So we have a virus, and it's coming in, binding to its cellular receptor. This is called attachment, where the virus adsorbs onto the host cell. Okay, because if a virus doesn't attach, it can't infect the cell and reproduce and make lots of little baby viruses. Okay, the next thing that has to happen is penetration. So if a virus can't penetrate, once it attaches, I mean, it could attach and just sit there, and sometimes that's what happens in certain situations, and then the virus doesn't infect the cell. But if a virus is going to be successful, it's going to penetrate the cell. And penetration can either be through receptor-mediated endocytosis, so we have kind of a capping phenomenon here, where viruses where the viruses bind to host cell receptors, and then capping occurs, and the virus is taken in like an endosome. But if the virus is enveloped, oftentimes what happens is the envelopes between the host cell and the viral envelope fuse. And when they fuse, then the capsid goes directly, the viral envelope kind of stays on the outside, and the capsid can go directly inside the cell. So the next step in viral replication is uncoding. So uncoding means that it gets the genome is revealed inside the cell. So the genome has to get rid of the capsid. It needs the capsid to protect itself once it gets outside the cell, but once it gets inside the cell, the genome has to be uncoded. So in this case, there's an example here of this bullet-shaped capsid coming off, and the RNA genome in this case is uncoded. And if the RNA genome is uncoded, and this is a positive sense, that means a host cell ribosome can hop on and start making viral proteins. Okay, so here's a couple of examples of uncoding within endosomes here. So this is a receptor-mediated endocytosis here. So we have a virus that's uncoding here in the endosome, and the capsid is left here in the endosome. And here's an example here of an adenovirus. So an adenovirus is taken in an endosome as well, but when an adenovirus replicates in the nucleus, and it doesn't uncoat, release its genetic material until it actually gets to the nucleus. So generally, RNA viruses will replicate in the cytoplasm, and DNA viruses will replicate in the nucleus. Generally, not all the time. Okay, so obviously a virus has to replicate itself. And when it replicates itself, that means the genome has to be replicated, whether it's positive sense RNA, negative sense RNA, which is antisense, double-stranded DNA, single-stranded DNA, it has to replicate itself. So here's an example where we had attachment penetration, now we're getting uncoding. So this is RNA that's coming out of the virus. Now we have a free positive sense RNA. This is kind of the simplest method here, positive sense RNA. And if it's positive sense RNA, it acts just like messenger RNA, so ribosomes can hop on. Ribosomes hop on and they start making a protein. And since this is positive sense, the first protein they make actually is an RNA-dependent RNA polymerase, because the virus doesn't carry a polymerase with it, because it can make its polymerase straight away. So then the RNA-dependent RNA polymerase goes in and it starts taking the genome and making negative sense out of the genome. So the negative sense is actually not the genome, but then the RNA polymerase turns back around and then can make a lot of positive sense. So if you're positive, you have to go through a negative. If your genome is positive, you're a viral genome that's positive, you have to go through a negative sense or an antisense intermediate in order to make lots more of your genome. And remember that when you're thinking about genome replication. Okay, so once viruses, so in the other slide, viruses were replicating their genome here in a vesicle, and in this slide, so we're taking a lot of these positive sense genome, and ribosomes are hooking up with it, and they're starting to make the viral proteins. The viral proteins, as we said before, are forming structures, they're self-assembling, and once we get a capsid starts to form, there's a protein sequence inside the capsid that attracts the nucleic acid for the virus, the RNA. And what happens is the RNA just slips inside the virus, attaches to that protein sequence inside the virus, and then the virus can leave the host cell, either by bursting the host cell or by budding from the host cell, which is what release is here. So the release is kind of, with an enveloped virus, release is generally budding. So viral proteins line up along the cell membrane, and somehow the virus, or the capsid of the virus, knows to translocate over where these envelope proteins are on the cell membrane, and the envelope proteins attach themselves to the capsid. So here's the nucleic acid inside this capsid, and then the virus can bud from the host cell. This is a picture of influenza, as a matter of fact. Okay, so the primary factor determining the life cycle of an animal virus is its genome. Okay, so you already know DNA viruses use host replication machinery because we're DNA, so we're made of DNA, and if a virus is made of DNA, it can, in general, it can use a DNA-dependent DNA polymerase. Okay, because we use DNA-dependent DNA polymerases to replicate our own genome, so why wouldn't viruses use the same thing? RNA viruses use a viral RNA-dependent RNA polymerase. So they either have to carry this along with them, or they have to make it as soon as they come into the cell. Because we don't make viral RNA-dependent, or we don't make any RNA-dependent RNA polymerases, because we don't have an RNA genome. We have a DNA genome. We make DNA-dependent DNA polymerases. And then retroviruses use a special reverse transcriptase, which turns their RNA, their negative sense RNA genome, into double-stranded DNA. So reverse transcriptase is actually an RNA-dependent DNA polymerase because it turns RNA into double-stranded DNA. Okay, so here's an example here where we have a DNA virus, papillomavirus. Papillomavirus, if you don't know, can cause warts. Okay, so biggest problem are genital warts. So here we have a double-stranded DNA virus binding to the surface of a cell. It gets it penetrates, gets translated or translocated to the nucleus. The genome is encoded in the nucleus. DNA replicates in the nucleus using a host DNA polymerase as well as a host DNA-dependent RNA polymerase to make its messenger RNAs, and gets translated. So this is much more like the human system, where we have a double-stranded DNA. Once the double-stranded DNA gets inside of a host cell, the host knows exactly what to do with double-stranded DNA viruses. And what that is it makes more double-stranded DNA as the genome for the virus, and then it makes messenger RNA, which makes the capsid proteins for the virus. So the virus really takes over the cell and doesn't really use anything of its own. It uses all the host proteins in order to replicate itself. So here's an example of picornavirus, which is poliovirus, is a member of Picornaviridae family. That is positive sense RNA genome. We've already kind of been over this, so a positive sense is RNA right out of the box. Okay, the first thing that's made from this positive sense RNA is an RNA-dependent RNA polymerase. So that's the first protein that's made, and then this RNA-dependent RNA polymerase can then make the intermediate negative sense, and it can also, then once it makes the negative sense, then it has a template for it to make more of the genome, which is positive sense. Okay, and proteins can also come off the positive sense because it's just like messenger RNA. Okay, so all viruses require a host cell for replication. I hope you know that by now. And they all face the same needs for infection. They all have to attach. So actually, if you're developing antivirals, you can look at each of these steps here, and you can figure out, if I prevent attachment, if I make an antibody to a virus and I neutralize the virus is not going to be able to attach to its host cell. And that's, in fact, a strategy that lots of people have pursued and been successful. Remember, antibodies are billion-dollar drugs. If you can stop penetration, if you can stop uncoding of the virus, if you can stop the genome from replicating, and when we talk about antivirals, we'll show how we can stop the genome from replicating by chain termination. If you can stop the virus from assembling, this is kind of a hard thing to do because we don't understand it very well yet, but we do understand how viruses can be released, and sometimes there's an influenza antiviral that we can use that suppresses release of the virus from a host cell. And if it can't be released, it just sits there and kind of decays on the host cell. So here's a clicker question. Why don't you pause your video and see if you can get the answer. So the answer is a capsid and a genome. All viruses have to have a capsid and a genome. Okay.